Hello, good afternoon. I hope you guys all had a restful reading week last week, and that you're ready to tackle some second half of the course. So as I mentioned at the beginning of the course, it is broken down into part one and part two, and we're done with part one now. Right? So you will not be tested on any of the things that you've learned previously. From now on, it's only going to be the second half of the course. And the structure is very similar to first semester. First half. It's very similar to first half in that we will have two quizzes, 10% each, one lab, another 10%, and the final test. Final test is going to be 20%. So in total, another 50% for the course. In terms of the format, it's all the same. Right? Same format for the tests. There will be some multiple choice, some multi-select, and sometimes there will be short answer questions as well. And the lab, of course, is going to be done online, but slightly different format this time around. Then I will explain it when we get closer to it. Okay. So in terms of difficulties, it can be a little bit more difficult in the second half, just because things are, there are more problem solving involved, and there are some concepts that are kind of abstract that you have to, it's harder for you to visualize. So make sure you continue to come to class, continue to ask questions, and hopefully is going to be manageable. All right. So here we go, lecture 6, cellular reproduction. So before we go into some of the mechanics of how a cell reproduce, there are a few important history lessons that we have to go through. Okay? And then these involves talking about various scientists who have made contribution to how DNA replicate themselves, for example, and the structure of DNA, as well as the fact that they figure out it was DNA that is responsible for passing on information to the next generation. So there are several experiments that we're gonna look at. Does anybody know what we're looking at here in this picture? What are these green little blobs here? Anybody know? Wild guesses.
The pen.
Okay, yeah, that's a good guess. Anyone else? Okay, so these are actually viruses. Okay, and they are made up of a protein shell. Okay, with some genetic material inside. Okay, so this here. Oh, I gotta change my pen color. This here, this yellow part here. This is a cell. Okay, and all these little hexagon-looking things. That's a virus. Okay. So let's take a closer look at what's going on here. Now, in the mid-1900s, although it was clear that the nucleus contains the hereditary information. When I say hereditary information, I'm talking about information that gets passed on to future generations. So it was clear that the nucleus has the hereditary materials in it, but the nature, the chemical nature of the hereditary material was unknown. I say that they are not sure whether it was DNA that's responsible for passing on genetic material or if it was the proteins. Okay? So on one hand, DNA has only 4 nucleotides, So it appears that they don't encode as much information as, say, proteins, because proteins have, 20 amino acids, right? As you know. But they have both DNA and proteins inside the nucleus, so they can't distinguish whether it's one form or the other. As you might recall, DNA actually wrap themselves around proteins to package in the nucleus, right? So, both of these things are in the nucleus, and scientists are kind of split, right, into two camps. One side thing is DNA, the other side thing is protein. And so there were two scientists, Hershey and Chase, seen here in these pictures. They did an experiment that helps determine it was the DNA that acts as a hereditary material and not the protein. So what I'm gonna do right now is to go through their experiment with you. Um, Strongly recommend that you have some colors with you because it is very complicated. Okay, so this is a Hershey and Chase experiment. Now, to understand their experiment, let's look at the basic structure of what's called a bacteriophage. Bacteriophage. This is a type of virus that infects bacteria. They are harmless to humans. They're easy to grow in the lab, and they are relatively simple. So the bacteriophage looks like what you saw on the picture. Okay, there is a shell, and then there is like a neck here, and then they have some legs. Okay, that's what they look like. For real, that's what they look like. I mean, like, I showed you the picture, right? Look, that's exactly what they look like with the legs and all that. Okay, so it's not like, you can't make this stuff up. So this shell on the outside, they as in the scientists, they know it is made up of protein. Okay, so there is a protein shell. Protein shell. And then in the inside, there are some DNA, DNA core. DNA core. So at the time, they know that the bacteriophage is going to infect the bacteria. They know something was injected into the bacteria. They didn't have like high power microscope back then. All right, so like the picture that we saw, that's like taken much later on. Okay, but at the time, they knew something was injected into the bacteria, and that something allows more virus to be created. So the question they try to understand, they try to figure out was it the protein that gets injected or was it the DNA that gets injected? Okay, so central question. What is injected into the bacteria? DNA or protein? Okay. In other words, is it the shell that went in or is it the DNA core that went in? And whatever went into bacteria, that's the stuff that is responsible for encoding the genetic material, because that's the stuff that allows more viruses to be created. Okay. So how did they figure it out? Well, they have two groups of bacteriophage. Two groups of bacteriophage. Number one, they are grown in sulfur 35. Okay, you might remember a long time ago when we first met, I told you about isotopes. These are atoms of the same element that have more neutrons, right? So that makes them heavier or lighter in some cases. If you remember from anybody remember from chemistry class, what's the mass of sulfur? Like if we look up the mass on the periodic table, anybody know sulfur? What's the number?
35.
35 is the number I have here, but what's the one in the periodic table? Anybody?
16.
Is it? No, I don't think so. Yeah, but good try. No. Do you guys have a periodic table handy? I'm asking because, like, it's gonna help you understand this a little bit more if you actually look it up. Okay, so look it up.
Yeah, I have sulfur 16 on the periodic table.
16 is the atomic number, yes, but what's the mass number?
Oh, okay, okay.
Yeah. Okay, Nathan just messaged and said 32.
32.0.
That's right. Okay, so like 32 point something, right? That means most sulfur is around 32. And this one is sulfur 35, so it's heavier. It's heavier. And again, I told you this before, I don't know if you remember. Heavier isotopes, they tend to break down. They tend to decompose on their own, and it gives off radiation. So the point is, you can detect where the radiation is coming from. Okay, so we have some bacteriophages that are growing in sulfur 35. So tell me, does the sulfur show up in the protein, or does the sulfur show up in the DNA? Which one contains sulfur? Is it protein or is it DNA?
Protein.
That's right. The protein contains traces of sulfur, right? So for the bacteriophages, for the viruses that are grown in the sulfur 35, their protein is kind of glowing. Not literally, okay? Like figurative speaking. Okay? The shell is glowing. Okay? And then they have the regular DNA. Okay? So grown in sulfur 35, the protein code is radioactive. In other words, you can detect them. Okay, you can detect them. What if you have to choose something that highlights the DNA and not the protein? What's a good element to choose that will show up in the DNA but not the protein? Do you remember? Think of the element that makes up DNA. Is there something that is only in DNA and not protein? Which one is it again? Sorry. That's okay. It's phosphorus, right? Phosphorus. Okay, phosphorus 32. Okay, so DNA has phosphorus, but protein does not. So in this case, the shell is not labeled. Yeah, the shell is not labeled, but the DNA are. So here we say the DNA core is radioactive. Right? So let's understand how this works. This is a bacterium. Okay, and then the virus is going to land on it. Virus is going to land on it. Something goes in, and then you have something that is left on the outside. Okay, we don't know which one went in and which one goes out. Okay? So something went in. And then what you can do is, you can put this into a test tube. Okay? And the test tube is gonna have solutions in it. And you can spin it down. You can spin it down. Okay? You can centrifuge. Separate the content. Separate using a centrifuge. Okay? Centrifuge is just something that spins really, really fast. It's a machine that spins really, really fast. And when you spin something really, really fast, the heavier stuff will go to the bottom. And then the lighter stuff is going to be at the top. So just by looking at this picture, right? Like, do you think the bacteria is heavier, or do you think the virus is heavier? What do you think, huh? Just like based on intuition.
Bacteria is heavier.
That's right. So what's gonna happen is we're gonna have two layers here. We're gonna have the heavier stuff. This is where the bacteria is going to be. Bacteria plus whatever was injected from the virus. And then on the top layer here, this is the part of the virus that that wasn't injected. Okay. In other words, this thing that's outside, this thing that remained outside, that's going to be on the top part. And then whatever was injected is now part of the bacteria, right? It's heavy together and it sinks to the bottom. You have to understand this concept before we move on. Otherwise, it's going to be like super confusing. So I'm going to take a moment here and see if you have any questions. Okay. Any question is fine. There's no stupid question. Okay. Like I'm sure you guys have some. Go ahead, please.
So,
Okay. We have. I'm sorry.
Go ahead, Mirhet. Yeah, Mirhet.
Okay, thank you. So if we have like a heavier one in the bottom bacteria, we say it is heavier, yes? So in the first one, is that lighter than the bacteria?
That's right. So the virus in general is much smaller than bacteria and therefore it's lighter. And when so like the virus has to attach onto the bacteria, okay? And then when you spin it at really high speed, the thing comes off. Right? So whatever was injected will stay in the bacteria, it will sink to the bottom, but whatever was left outside will now separate and go to the top.
Okay, thanks.
Yeah. Fatima, you have another question?
Yeah, I had, so I got my answer right now because my question was, if the virus is still attached to the bacteria, so how come they get separated when it's like spinning? Yeah.
That's right. The spinning is really fast, right? Like they cannot hang on and they come off basically. Okay, very good.
Okay, Vincent, so in the test tube, do we have like a liquid or
yes.
like,
there's a liquid in there. Okay, it's not like because I didn't know this was a problem for many people until recently. Someone told me they are asking how come the top part doesn't fall down to the bottom. It doesn't, because it's like this is like salad dressing. You can think of, you leave the salad dressing in the fridge, like the oil is gonna flow on the top, right? Like the oil is not gonna sink down, right? So like this whole thing is like filled with liquid. The heavier stuff go to the bottom, and then the lighter stuff goes on top. Okay. All right. So this is what they did. Okay.
So, sorry, I have a question.
No problem.
So we already put a solution in the in
the test tube.
Test tube.
Oh, okay. No, again, this is so like how do you put the virus with the bacteria, right? Like in reality, you have bacteria in a solution, right? Bacteria.
Oh, yes.
Yeah, and then you introduce the virus, and so it's already like all floating in a soup, if you would. Okay.
Okay, thank you.
Very good question. Okay. So let's see what's going on here, right? Let's take a look. They took the group one virus. Okay, group one. The group one here are the one that have the glowing protein. Okay, group one, glowing protein. And in other words, it's radioactive. Okay, the code is radioactive. And then they let these viruses infect the bacteria, and then they spin it down in the test tube. They spin it down in the test tube. Now, you and I hope we already know it is the DNA that is responsible for the hereditary material, right? DNA is the genetic material. We know that already. That's what we've been learning. But they didn't know back then. That's why they're doing this experiment, right? So, knowing what we know, I want to ask you, do you expect the radioactivity, the glowing yellow thing, to be at the top or to be at the bottom? Knowing what we know now, where
At
do
the
you
top.
expect? At the top, exactly. Because the protein is not the genetic material. We know it. They didn't know, but we know it. So that means the protein didn't go in. The protein did not go into the bacteria. And so the radioactivity was detected at the top. Okay. So what does this tell them? They're like, okay, we only label the proteins because we use S35. We only label the protein, and we only detect the radioactivity at the top. The top represent the part that did not go in. Okay, so conclusion here, conclusion. Protein, the virus protein, okay? The virus protein shell stayed outside. Stayed outside. Now this is a little bit tricky. This only tells you, let's stop writing, listen for a second. Huh? This only tells you the protein did not go in. It does not tell you that the DNA went in. I have no idea where the DNA is in this setup. Maybe the DNA didn't go in either. I have no way of tracking the DNA because the DNA is not labeled, right? Okay. I cannot label both of them at the same time. If I label both of them at the same time and something is glowing, I don't know if it's the protein that's glowing or if it's a DNA that's glowing, right? Only the protein is labeled here. So if it glows, I know it was from the protein. And because only the top portion is glowing, I know that's the one that did not go in. Questions? Okay. So what about group 2? What if they use the group 2? In the group 2, the shell is not labeled, but the DNA is labeled. Right? So once they spin it down, we would expect the radioactivity to be at the bottom. Okay, conclusion. Virus DNA core, DNA essentially, went into the bacteria. Okay. So taken together, the final conclusion. DNA, not protein, is the hereditary material. Okay. So Hershey and Chase were the first scientists to show that DNA is the genetic material. It's the hereditary material, the stuff that get passed on to the next generation. Are we good?
Vincent, so for the final conclusion where they said, okay, DNA is the hereditary material, is it because of the radioactivity, or like what gave them the, like the idea that it's DNA and not the protein?
Okay, because the protein, as the experiment showed, the protein never went into the bacteria, right?
Oh, okay,
Yeah. The
okay.
DNA went in. Before they did this, they knew whatever went into the bacteria is responsible for making more viruses.
Okay,
Yeah.
okay.
So in general, right, for those of you who don't know this, the virus inject the DNA into the host, the bacteria, or it could be a human cell, and then it hijacks the ribosomes and all that stuff to make more viruses, right? And so they knew about this. They knew about the hijacking and stuff. They just don't know what was hijacking it. Was it the DNA or was it the protein? Turns out it's the DNA. Okay. So there you have it. That is Hershey and Chase. I hope you found that all right. Okay, this is just like first of several experiments that we're going to be looking at. And again, right, just go through it again after class. And
Professor, excuse me.
yeah.
Hereditary material means like a DNA? Is that DNA?
Hereditary material means things that is inherited from previous generations, like it get passed on, right?
Okay.
So for example, like, if I have brown eyes, I can pass that on to my kids. They will also have brown eyes, right?
Okay,
So
yeah.
that's hereditary material. Okay, that is that. We'll come back to this in a bit. There is your picture. You can actually see the DNA. Look, this line that's going in, that's the DNA that's being injected. Okay, so this is for real. And then these are the virus shell, the empty shell that had finished injecting the genetic material, and then they just kind of break off as a shell. Okay. Okay, we did that already. This is just to show you what I already wrote down for you. Okay, nothing new here. This is the professional drawing that shows you what happens if you label the shell and what happens when you label the DNA. Okay, you see the shell stays outside and the genetic material went in, right? All right, what's next? After Hershey and Chase experiment, people now accept that it was DNA that's responsible for passing on genetic information. So the next thing that the scientific community was trying to solve was the structure of DNA. Now that we know this is important molecule that determines, the various traits of an organism, what exactly does it look like? So the work was started by Rosalind Franklin. She used X-ray in something called X-ray crystallography to study the structure of DNA. And she came up with the idea that the DNA is a double helix. So that's what like when you shoot X-ray at crystallized DNA, that's the diagram, that's the picture that she got. And based on that, she was able to deduce that DNA is a double helix structure, the one that we are familiar with, right? Like the twist. But unfortunately, she died of cancer before she was able to finish her work. Probably because she was working with X-ray a lot, and she didn't. Back then they didn't really have a lot of protection, I suppose. And so Watson and Craig, which are these two fellows here, they continued to work on her data, and they were the first to come up with a scale model of the DNA double helix. And in every textbook where you see Watson and Craig, you will almost always see this picture. Okay, double helix. They went on to win the Nobel Prize, I think. But many people say it should have been Rosalind Franklin who would receive the Nobel Prize because she was the one who came up with the data in the first place. So in any case, you know, by this point, people know DNA is the hereditary material. They know the structure is a double helix, okay? And then the next thing that people wanted to figure out was how DNA copy itself. Okay? Being the genetic material, they know there has to be some ways to copy the DNA so that you can pass it on to future generation. Okay? So that is the work of these two people, Messelson and Stahl. Messelson and Stahl. But first, let's remind ourselves what is the purpose of DNA replication. So before a cell divides, it has to copy all of its genetic material, all of its DNA. So let's say we have 2 chromosomes here, right? 2 chromosomes. We actually have 46, as you might recall, but for simplicity, there are 2 here in this cell. So you copy all of them, okay? You get 2 of each. So when you separate, you get 2 and 2 again, right? Each of the daughter cells, that's what we call them, daughter cells, each of them are genetically identical to the parent cell. It's the same. Okay? And in order for this to happen, you have to copy all the genetic material, all the ACTGs you have in your DNA, all of them must be copied and be passed on to the next generation. Okay? So the problem here is they don't know how this is done. How does DNA copy itself? Well, there are 3 models here. Okay. First, we have the semi-conservative model. Semi-conservative model. So think of the pink ribbon as the original genetic material. And you separate the 2 strands, and then you make a new strand here. Okay. The blue one represents the new material. Okay. Never mind the second cycle for now. We'll talk about that later. So that's the semi-conservative model. You just pull apart the 2 strands of DNA, and then you build the missing half. We know that's possible because DNA strands are complementary, right? If you know half, you can build the other half. If you see A, you know you have to put a T next to it. If you see C, you know you have to put a G next to it. So this is called semi-conservative model. The conservative model suggests that you do not pull the strands apart. You just have the original, okay, and then you make a carbon copy of it. You make a copy of it. Okay, so entirely original strand, entirely new strand. And for dispersive model, it's a mixture. Yes. In the same strand, it's both new and old material. Right? As opposed to semi-conservative, one strand is original material, the other strand is new material. Right? Here, the new and old are kind of interspersed within each other in the same strand. Now, I understand this is very hard to visualize. Okay, so I made this picture. It took me 30 minutes to make this. Okay, so I hope it is going to be helpful for you. So let's say we have an original picture here. Okay, teddy bear, right? And you can photocopy it like this. And you can just put it in the photocopier and then you make a copy. Do you think this is conservative, semi-conservative, or dispersive? What does that look like to you?
Conservative.
That's right. This is conservative. Okay, conservative. Right? You end up having 2 copies. One is completely original. The other is just a copy of it. Over here, you know, you photocopy it, but then you know you kind of rip the paper in half, and then you stitch the original and the copy together to get one thing, and then the other one you get the other half, right? And what does this model seem like to you?
Semi-conservative.
Semi-conservative.
That's right. This is semi-conservative, right? And then this one This one took the longest to make, okay? But it's like patchwork, right? Some parts are old, some parts are new, and this is the dispersive. Okay? So there are 3 separate models. They don't know which one is correct. That's why they have to do an experiment to figure it out, right? So I'm going to show you the experiment now. This one is very, it's, you know, it's hard, right? But I'll try my best. Okay, I'll try my best. So here we go. Okay, this is called Michelson and Stahl experiment. Michelson and Stahl. All right. So this is how it goes. Okay, They worked with E. coli. E. coli is a bacteria, okay? It grows very fast and they divide very rapidly. And so every time it divides, it has to copy all the genetic material, right? So this is what they did. Okay, they grow. I'm just gonna use present tense. Okay, it's much easier. They grow E. coli in heavy nitrogen. Heavy nitrogen. Okay, we will call it N15. Please use the same color as me. If you do this in black and white, it's not gonna make sense. Okay, you have to use some color here. Hopefully, you have a red pen and a blue pen. So grow E. coli in heavy nitrogen, N15. The normal nitrogen is 14, okay, if you look it up on the periodic table. So grow E. coli in heavy nitrogen for several generations. Okay. If you grow them in several generations, then all the nitrogen in the DNA contains N15. Okay. Most of the nitrogen, okay? It's pretty much all, okay? To keep it simple, just think, because that's the only thing that's available. To make new DNA, they need to incorporate nitrogen, right? If you only give them heavy nitrogen, that's the only thing they could put in their DNA. And after several generations, all their DNA will be heavy nitrogen, heavy DNA. Okay. So if you extract all the DNA and you put it in a test tube, again with liquid in it, and you spin it really high speed, where do you expect the N15 to be? Where do you expect the DNA to be? Is it at the top? Is it in the middle? Or is it at the bottom? What is it again? Do you guys understand my question?
Bottom.
Bottom? Yeah, that's right. You're gonna find the N15 right here. Okay, this is where the heavy N15 DNA is gonna be. Heavy things goes to the bottom, right? Heavier things sinks to the bottom when you spin it down in a test tube. Okay, so remember, this test tube has some liquid in it, okay? And then, you know, the heavy DNA is going to sink to the bottom. Okay. When DNA are extracted and separated and spun down in a centrifuge, N15 DNA sinks to the bottom. Okay, there was not a lot of answer when I asked where the N15 is gonna be. So, do you guys have any questions you want to ask to, clarify this before it gets more confusing? Because it will get more confusing, I'm sorry.
So, Vincent, what is the purpose of doing this? Like, what do they search for?
Okay,
What did
good.
you
Yeah.
say?
So the question is, what model? What is the correct DNA replication model? Is it conservative? Is it semi-conservative? Is it dispersive?
Okay, Vincent. So this is when they've already agreed with Rosalind that it's helix, right?
That's right. So this is already, they already know DNA is genetic material. They already know it's a double helix.
Okay.
But they don't know how to copy it. Like, do they separate the helix and then make new copies? Do they just copy it directly? Is it like all over the place? The three models, they don't know which one.
Oh, okay.
All right. Okay, we haven't talked about the experiment yet. Okay, I'm just giving you the background information. Are we all clear on why, if you grow the E. coli in heavy nitrogen for several generations, and then you spin it, you take the DNA out and you spin it down in a test tube, the DNA will be at the bottom. Do we understand why that is the case? Why is it in the bottom and not like, at the top? The answer is because it's heavy, right? Because it's heavy. Heavy thing sinks down in a test tube. That's basically it. Okay. So what do they do next? What they do next is this. They transfer the E. coli, the one that had been growing in heavy nitrogen. They transfer E. coli to N14, to light nitrogen. When I say light, really, it's like the regular nitrogen. That's the N14. For one generation. And extracted the DNA. Right? And extract the DNA. I forgot to use present tense. Extract the DNA. So let's think this through. Okay, let's think this through. We have the original. Let's call that generation 1. Generation 0. Okay, generation 0. Okay. That's the one that had been growing in heavy nitrogen for several generations. So what does their DNA look like? Well, as you can imagine, it's just like this, right? And, with the letter. This is N15, N15. Both strands are N15. They are very heavy, okay, because that's all they have. Please use color. Okay, otherwise it's not gonna make sense. So let's take a look at what happens to the first generation. Okay, this is generation 1. After one division, when you have transferred to the lighter DNA. So what if it's conservative? What if it's conservative model? Well, if it's conservative model, then you're gonna have the original DNA, N15, N15. And then the new one that you're making is all gonna be new material. What kind of new material is available? Only the N14, right? So you're going to have this. N14, N14. Do you understand what I'm trying to explain here? It's okay to have questions.
I don't have a understand because why it's N14.
Say that
And
one more time. What's the question?
the
Why is it
question,
N14?
yes.
Okay. So you grew them in N15 for several generations. You understand why everything is N15 at this point, right?
Yes.
And then they move it to the N14 for one generation. So that means at that point, when the E. coli divides, when it makes new DNA, anytime you make new DNA, you need nitrogen, right? And the only available nitrogen at this time are the N14s. Okay? So if it's the conservative model, then the DNA that you have in the culture would have N15 from the previous generation, and all the new ones that you've made will be
N14.
N14.
Yeah.
Okay.
Yeah.
So listen, everybody, if you put this in a test tube, right, if you collected all the DNA and then you put it in a test tube and you spin it down, okay, remember it has liquid in it, right? How many DNA bands are you gonna see?
2.
2. That's right.
2.
Okay. You will see, and I hope you're using color. Okay. You will see the heavy one at the bottom. Okay. N15, N15, and you will also see a lighter one at the top. N14, N14. Okay. That's the conservative model. Okay. This is just a liquid at the top, okay? If it confuses you, just erase it. There. Let's go. Okay, this is a test tube, huh? All right. What if it is semi-conservative? What if it's semi-conservative? What does it mean? Well, if it's semi-conservative, it means you're gonna have to separate the DNA strand. And separate them. And then you will build the other half, right? Using complementary rules. A goes with T, C goes with G. But I only have N14 available. So everything is going to be blue on the newly created strand. Okay. I'm not going to draw the lines in the middle. Okay, it's too hard to be half blue, half red, but you get it, right? I hope you get it. So this is N15/N14 in the same DNA molecule. Same thing over here. So if you spin this down, if you collect the DNA, spin it down in a test tube, how many bands do you expect to see here? And this requires a bit of thinking here.
1
Where would that be, Fatima?
in the bottom.
1 band at the bottom?
Yeah.
Okay, anyone else?
The middle?
The middle, that's right. Okay, you're gonna have something in the middle. I'm just gonna use purple here, right? You're gonna see a mixed band because look at the DNA. It's not as heavy as 1515, is not as light as 1414, right? If anything, it's like 14.5, right? It's somewhere in between. And that's what you will get. Okay, you will get a band that is in the middle. And that band is a combination of 14 and 15. Does that make sense, Fatima? Since you said
Yeah, I can understand now. Yeah.
good. Okay.
Excuse me.
Jane, go ahead.
I can't understand where does the 14 nutrient come from.
You don't understand where the 14 nitrogen come from?
Yeah, because we doesn't have it doesn't have 14 nutrient in the
In the generation 0?
Yeah.
Yeah, okay. So generation 0 only N15. You understand that part, right?
Yeah. Only
Yeah,
15.
and then they took that bacteria and then they put it into the N14 before they allow to split. Okay? So once they put it in N14 and then they need to split, from that point on, between generation 0 to 1, the only thing that they could put in the DNA is the 14. That's where it came from. Okay? So look, you have a bacteria here with the DNA in it, okay? That is generation 0. And then when it's split, you give them N14 only. So when you get to generation 1, this is generation 1.
Who give the human, the person?
Oh, who put them in N14?
Yeah.
Oh, the people who are doing the experiment, mesosynthesis stuff.
Oh.
Yeah, that's right.
Oh.
So yeah, so like someone has to move the bacteria. They didn't just move themselves. Yeah. Any other question? These are great questions. Anyone else?
Okay. Thank you.
Yeah. Anyone else? Okay. So next is dispersive, right? Dispersive. And in dispersive, this is really hard to draw, but it's basically, a bit of red and blue, but not restricted to the same strand like semi-conservative. They're just like all over the place, right? Okay, all over the place. Like that. And it is also a mix of 15 and 14. And so when you collect the DNA, when they collected the DNA and they spin it down in test tube, how many lines are we expect to see? How many bands? How many DNA bands? 1, 2, 3. How many? It's not a trick question. Just go with your gut feeling. It's 1 band, 2 bands, or 3 bands.
2 bands.
2 bands. Okay. Anyone else? Let's do a vote. Okay. Let's do a vote.
1 band.
How many bands? Okay. 1 band A, B, C. Okay. Not sure is okay as well. My question is, how many band are you going to see?
It's 1, sir.
Yeah. Okay. Okay. I got half of the class response. It's gonna be 1 band, and it's gonna be in the middle again. Because look, this DNA consists of also half of it is 15, half of it is 14, right? So it's not as heavy as 1515, is not as light as 1414, is somewhere in the middle.
Excuse me, Professor. So what is the difference when both are the same, semi-conservative and dispersive? Because
That's
I
right.
see
So
the result, everything the same.
okay, not everything is the same. The band is the same. Where you find the band is the same. But then the structure is different, right? Like maybe I go back to my teddy bear's picture here. Semi-conservative is half old, half new, but the old is on one side only, and the new is on one side only. Dispersive is also half old, half new, but the new is not restricted to one side. It's like all over the place.
Okay, thank you.
Yeah. So if you look at the picture here, right? Semi-conservative, like the whole thing is either red or blue, red being original, blue being new. And here, each strand is a mix of new and. Okay, so this is generation number 1. And then what happens? What happened? Well, this was observed. This was observed. And this was not observed. Okay, so after one generation, they observe, they see, we'll just keep it simple. They see only a single band. Therefore, the conservative model is incorrect. It's not correct. If it's the conservative model, they should have seen 2 bands. They did not. They only see 1 band. There was only 1 band. So that means it's either semi-conservative or dispersive, right? But they can't tell which one it is at this point. Okay. But for sure, it's not conservative. They did not see 2 bands. So what do? They grow E. coli, in N14, N14 for another generation. Okay, they're going to do this one more time. So let's see what happens if they do this for another generation. Okay. So generation 2. Well, if it's a semi-conservative model, we will separate the strand. So I will have the red one, and then I will have the blue one. This blue one is from the previous generation, right? Okay. And now we have to build another complementary strand. What's available? Only N14. So it's going to have blue strand here, blue strand here. Same situation here. Same situation. You will separate them. And then you will build the blue strand. So if you put it in a test tube and spin it down, help me out here, please. How many bands are we gonna see?
2.
2. And where is that, Fatima? Top, middle, bottom, where?
The N15 goes to the bottom, and the amount is less than N14, which goes to the top.
Okay, but there is no N15, is there?
Yes, because it came from generation 1.
Oh, no. This is only generation 2. No. Okay, I see what you mean. Okay. No. Yes and no. Okay. This one here is one DNA. The DNA is not gonna separate. Okay. The strands are not gonna separate.
Oh, okay.
Yeah. So try again. Where is it gonna be? Where are we gonna see it?
On the top.
The low is the bottom. The
Okay.
N15 and 14 will be at the middle.
That's right.
And the
N15
and 14
at the what? You got cut off at the very end there.
at the top.
At the top. That's right. Okay, so this is the 1414, and then you will have the 1514 in the middle. Okay, look, that goes to the top, this goes to the middle, right? It doesn't go to the bottom, because the bottom is only for when both of them are 15, right? And that doesn't exist.
The bottom's gonna be water. Sorry, I don't know.
Okay, so like this whole thing is like a gel. It's like I know it keep on saying liquid, right? But it's more like a syrup type of thing. So that's why you know, things will go to their particular location and stay there. Because you know, if you think of like syrup, right, it's very thick, right? Like, so once they go to that place, they're gonna stay there. It's not gonna, like, you know, sink down or flow up or anything like that. So at the bottom, it's just like it's not air, okay, but it's like empty gel. Does that make any sense? It's really hard
Yes,
to,
yes, thank
yeah.
you.
Okay. Yeah, and that's what people were having trouble understanding. It's like, wouldn't this one just fall down because of gravity? No, it's like it's gel over there. Okay. All right. If it's dispersive, then okay, I'm just gonna cheat a little bit here. If it's dispersive, it's the exact same thing. The exact same thing. Right? You're gonna get more of the same thing here. And how many bands do you expect to see in this case? How many bands in generation 2?
One in the middle.
Yeah, one band, right? One band in the middle. Okay. So they did the experiment, and it turns out, oh, this is not working. 14. It turns out that they saw this one. Okay, they saw this one, and not this. Okay, not this. Okay. So conclusion. Correct model is semi-conservative. Semi-conservative. All right. Okay, I know that was a lot to take in, but you know, go through it again. Okay? Email me if you have questions. Okay, let's do some questions, poll questions here, and see if you understand it. Go ahead. Okay. The correct answer is to label the protein coding of the virus. okay? The outside shell, right? So that you can keep track of where the protein goes. Okay, this one is probably quite hard. I'll just tell you the answer, okay? So this is saying if DNA replication follows the conservative model, that's not true, right? I just show you it is the semi-conservative model. But this is saying if it did follow the conservative model, what would be the expected result? So we go back to here. We go back to here and we see that the conservative model is in the middle. So if it was conservative model, we would copy it. This is first generation. You would see two bands. You would see a band at the bottom and a band at the top, right? Nothing in the middle because we don't have something that's mixed, right? And then if you do it for another generation, you would see a band at the top again that represent N14, N14. And then you would see a band at the bottom, right? This band at the bottom came from the original, right? So you will see two bands in both generation 1 and generation 2. So the correct answer for this question would be A. Okay. This here, this is dispersive. Right? I showed you in the notes. And over here, these actually don't make any sense. Well, this one does. Okay, this one is a semi-conservative. Don't get too hung up with the color. Okay, like the color that is being used in this question is opposite to what we use. Like don't get too attached to the color. Color doesn't. It's not the important thing here. What's important is the location of the band. Like, is it at the top? Is it at the bottom? Right?
Vincent, I have a question.
Yeah.
Can you go back to the notes, please?
Sure. 1 second. Yep.
Can you slide to the left, please, for the conservative?
Okay. Yeah.
For the conservative, you said that it formed two bonds, right?
It forms two bonds.
With your drawing, the test tube drawing, there's two bonds on it.
You mean the lines in the middle?
Yes, the lines on the
Oh, I mean they all have it.
in the test tube.
Sorry. Two bands. Yes. There is one at the top for the N14, because that one is only light nitrogen, right? And then the one at the bottom is only heavy nitrogen. You only have a band in the middle if it's a combination of 14 and 15.
So why does it say in your note that after one generation they see only a single band?
They did. Okay. Oh, I see where the confusion
So
is.
sorry again.
No, it's okay. So after one generation, they only see a single band. This one generation. This here is referring to like the whole first generation. So I guess your confusion is like you might thought this applies to the conservative, but no, I'm saying
That's correct.
yeah, it's like a general statement. Okay, like after one generation, they only saw one band, so it only could have been the semi-conservative or the dispersive, right? They did not see two bands. That's why they rejected the conservative model, right? That's what I'm trying to tell you.
Oh, I see. Thank you.
Okay. Yeah. All right. I don't think I asked you like there's just like one or two questions on this on the quiz, okay, or test. If you understand like this one, you should be okay. Again, don't get too hung up with the color. I'm trying to change it into black and white, but I don't know how to do that. Oh, there it is. Okay. Yeah. So I changed it to black and white now, sort of. Hopefully it'll make more sense. Oh no, I'm just making it worse. Okay, I'm just gonna leave it as it is. Okay, that's all. We're gonna skip the break and then I'll let you go at 2. Okay. Just want to go through a few more things here. So the next thing we're gonna learn, it's gonna take another hour, which we don't have. Right. So I'm just gonna set up the groundwork for you, so that when we come back on Wednesday, you will we will be able to build on that. Okay. Mechanisms of DNA replication in prokaryotes. Okay, remember prokaryotes are simpler organisms, like bacteria. They are prokaryotes. They don't have a nucleus. We are eukaryotes. We have a nucleus, right? So scientists have a good understanding of the replication process in prokaryotes, primarily from studying E. coli. Again, they're easy to grow. They divide very rapidly, so you can observe it readily. And it turns out the eukaryote replication process is very similar to the prokaryotes, okay? But just a little bit more complex. So to start you off, I'm going to show you what happens in prokaryote DNA replication. Okay, like how do the bacteria do it? And then from that, I'm going to show you the human versions. So the parent molecule unwinds, and then the 2 new daughter strands are built based on base pairing rule. I kind of already alluded to you, right? Like we start with the original DNA strand like this. Okay. You know what? Actually, I can make it more visually clear to you. Okay? It's up to you if you want to copy this or not. Okay, this is like the original DNA. We call this the parent strand, the parent molecule, parent DNA. And then what happens is you will separate the 2 strands, and you're gonna build a new one. So your body sees A here is okay. It's gonna go with the T. T goes with the A. C goes with the G. So on and so forth. Same thing here. When it sees a T, it knows to pair with an A. Using the base pairing rule that we learned, we end up having 2 strands, right? Each strand here is called a daughter strand. Daughter DNA, and it's going to be identical to the original, identical. Okay? And you get 2 of them now instead of 1. This process is really, really fast and very, very accurate. For prokaryotes, for prokaryotes, I mean, you don't have to memorize these numbers. I'm just like telling you, okay? For prokaryotes, it's like 1,000 bases per second. Okay? Every second is able to copy 1,000 ACTG. Think about your typing speed, right? How fast do you type? How many words can you type per second? Okay, I don't know, 3 or 4, right? That's already pretty fast, right? So the prokaryotes are able to copy 1,000 letters per second, essentially. Okay? And in E. coli, there's approximately 4 million base pair. 4 million bases in E. coli. So, if you do the math, if you take like 4 million, okay, I'm keying into the calculator, 4 million divided by 1,000, that takes about 4,000 seconds. Okay, so like a little less than an hour, you can plug it in yourself, right? 1.11 hour, it's able to copy all of its genetic material. For eukaryotes, that's us, okay? Humans, eukaryotes, we are a little bit slower, but still quite fast. Okay, 40 base pair per second. And humans, we have approximately 3 billion bases Right? So a lot more to copy. We're a little bit slower, but slower is okay. We don't want to make mistakes, right? Because you make mistakes, it's going to have a mutation, and that might or might not lead to things like cancer, which is something we'll talk about, I think, in this lecture. Yes, we will talk about this in this lecture. Okay. So the process involves more than a dozen enzymes and other proteins to make it happen smoothly. Okay, so here is a list of enzymes. Okay, we have something called helicase, gyrase, all these things. Right? And then we're gonna have to draw this out next time, and I'll give you more information about what each one of them is responsible for. But yeah, sorry, go
I
ahead.
have a question. Like my question is like a general question. So I just want to know all and each cell in one human body, like disregard like the task like heart cell, brain cell, and everything. So do they all have same DNA?
That is excellent question. Okay, and that's something that people often have a hard time visualizing. Okay, so this this is actually something we're going to talk about. But now you raise the question, maybe we can talk about it now. All the cells in your body all have the same set of DNA. Okay? We have 46 chromosomes. 46 chromosomes. Okay? And chromosomes, as you know, are just very condensed version of DNA. Okay, so we get 23 from mom, 23 from dad, from the sperm and the egg, and then there are the 46 chromosomes in the body, right? So all the cells in the body have 46 chromosomes, except for the sperm and egg. Okay, in female, like the eggs would have 23 chromosomes. In men, the sperm would only have 23 chromosomes. So that when we reproduce, the sperm and the egg come together, and then the next generation has 46, right? But let's forget about the sperm and egg for the moment. Every cells in the body, the heart cells and the brain cells and the liver cells, muscle cells, all of them, each of those cells would have their own 46 chromosomes. Now, do they all use all 46 chromosomes? The answer is no. Okay, we'll learn more about genes later on. But the chromosomes is like a set of instructions for the cell to do different things, right? And even though you have the whole set of chromosomes, the chromosomes that gets the parts of the chromosome, I should say, the parts of the chromosome that get used is going to be different in a heart cell than it is for like a brain cells. Okay, so think of them as like on-off switches. Each chromosomes have a bunch of on-off switches. You don't turn on everything in chromosome 1. You only turn on what you need in a heart cell. The rest of it, you turn it off. And then in a liver cell, same thing. You might turn on some of the similar genes, okay, but then you might switch off other ones. So even though they all have the same set of chromosomes, not everything is going to be activated at the same time. Not everything's going to be turned on. Okay, think of like you and I, we both might have the same set of I don't know, like encyclopedias at home or something. Okay, but like what you use is very different than what I use. Okay, we might use different pages, for example. So that's kind of like that. They have the same library, they have the same collection of information, but what gets switched on and off is unique for each type of cell. Does that answer your question?
Yeah, very good. Thank you.
Okay, so we're gonna look more into that later when we learn about genes and proteins and in fact, what really scary is sometimes if you turn on something that's not supposed to be turned on, then that could lead to cancer, right? As we will see later on in this lecture. So like I said, this note is gonna take at least 45 minutes, if not an hour to complete. I don't want to start now and not be able to finish because if you do something and then you leave it for one day and then you come back, you're just gonna forget stuff, right? So I think I'm gonna stop here today. You already have a lot to go through on your own, and then Wednesday we will resume. Okay. Well, thank you very much. Enjoy the rest of your day. Bye bye.
Thank you.
Vincent, can I stay with you? I have a question
Sure. Yeah.
personal
Yeah. Go ahead.
about my program
Sure. Sure.
after